Welcome to the Histology Lab. Histology is the study of disease in organs, tissues and cells. Here at Leicester, we receive around 65,000 specimens per year. These specimens come to us from surgery within the hospital, from day-case clinics, from GP clinics in the county and also private clinics as well. The first stage in this journey is receiving the specimens. We receive specimens throughout the day, and as soon as they're received, they are sorted and allocated a unique patient identifying number. All of the specimens that we receive in the department are dissected by a pathologist or a specialist biomedical scientist. This specimen shows a gallbladder. You can see the green appearance because it's filled with bile. Bile is produced as part of normal function in fat metabolism. The specimens are dissected in a specific way so that we can look at the areas of interest within each tissue type. The parts of the specimen that we want to look at further are placed into cassettes which have the unique patient identifying number on them. We receive a whole range of different tissue types, from very tiny biopsies which are only 1 to 2 millimetres, right up to whole organs and resection specimens. The biopsies are used to make a diagnosis. The resection specimens are used to treat the patient. We receive both malignant resection specimens and benign resection specimens. We receive samples from every part of the human body. 90% of them are from live patients. At the end of the day, when all of the specimens have been dissected, the blocks that are produced are then placed onto a processor overnight. Human tissue is made up of around 70% water. We need to remove this water and replace it with paraffin wax to allow the tissue to have enough structure for us to cut a section from it. This is what the processing machines do. They use a series of graded alcohols overnight and then wax. So in the morning, all of the specimens are ready for the next stage of the process. The specimens are removed from the processor and then taken through to the diagnostics lab to be embedded into paraffin wax. We embed around 500 to 700 blocks by hand per day. So each cassette is removed and the lid carefully taken off. We choose an appropriate sized mould to put the tissue into. A small amount of wax is placed into the bottom mould. The tissue is then carefully transferred from the cassette into the mould and orientated in the correct way. The tissue is then transferred onto a cold plate and pressed down so that it's flat. The cassette is placed back on the top and the back of the mould is filled up with wax. These are then placed onto a cold plate until the paraffin wax is set completely. When the wax is completely set, it's removed from the mould, and here you can see the tissue that's embedded in the paraffin wax. The excess wax is removed from the back of the blocks, ready for the next stage in the process, which is microtomy. Microtomy is the process that allows us to cut a really thin section of tissue and mount it onto a glass slide. We used a specialist piece of equipment called a microtome to allow us to do this. We used surgical stainless steel blades, which are inserted into the microtome. The block is then placed into the chuck, and as the handle is turned, every time the block passes the blade, it removes a thin slice of tissue. The first step in this process is called trimming in, and this allows us to remove all of the excess wax from the top of the block so that we can see all of the tissue that we need to be able to make a diagnosis. When the block's been trimmed in, it's placed onto ice to get cold. Then once it's cold enough, we can actually take a section of tissue from the block. We routinely cut all of our sections at 4 microns thick. When we have a ribbon of sections, the next thing that we do is to float it out onto a water bath. The water is set just below the melting point of the wax, and this allows the section to flatten out. We can then pick up this section onto a glass microscope slide, and it's labelled with its unique patient number. All of our 500 to 700 blocks are cut by hand per day. Once the blocks have been cut, the slides are mounted onto our staining racks to go onto an automated staining machine. Here you can just about make out an unstained section on every slide, but if you looked at this down a microscope, you wouldn't be able to see any of the detail or structure of the tissue, so you wouldn't be able to make a diagnosis from it. All of the slides that we produce are stained with hematoxylin and eosin stain. This is the international reference stain that is used in all histology labs throughout the UK and worldwide. The hematoxylin stains all of the nuclei a dark blue colour. The eosin stains up all of the other parts of the tissue a pale pink colour. This then allows our pathologists to be able to look at the individual cellular components to be able to make a diagnosis. This shows our automated staining machine taking it through all the different stages required. A cover slip is then placed onto the slide, and then every slide needs to be quality checked before it can be booked out of the department. One of our specially trained biomedical scientists checks the quality of the staining and of each section produced from each block. We microscopically quality control specific tissues. For example, skin specimens, we need to be able to see all of the epidermis of the skin. So our biomedical scientists will check these using a microscope before they leave the lab. Then the final stage in the process in the lab is for the specimen to be booked out, and this is where it's allocated to the pathologist for reporting. So please watch the next video, which takes us through the reporting process, where you can actually see some of the case studies that we do.